So today I would like to talk about microbial communities and how we can use the information that's contained within them to get a better understanding on how we can convert lignocellulose into useful products, as well as for important in nutrition. And so because I'm going to talk about microbial communities, I'm going to talk about my favourite quote from a microbial ecologist. This is Tom Curtis at the University of Newcastle. So Tom said that he makes no apologies for putting microorganisms on a pedestal above all living things. For if the last blue whale choked to death on the last panda, it would be disastrous but not the end of the world. But if we accidentally poison the last two species of ammonia oxidizers, that would be another matter. So the point of this quote is to illustrate that the vital roles that microbes play in the natural communities and the influence they have over all walks of life on Earth. So as Vincent said, I'm a member of the larger PEP group at NMBU, and so all the vast projects that are contained within that group focus on the bioeconomy. So these are the parts of the economy that use renewable biological resources from land and sea. And in particular, we focus on projects that look in towards improving bioethanol production, improving recycling organic waste into biomethane, and to understand the processes that occur from digestion of feed and important production animals here in Norway. So one of the bottlenecks that confronts all these three aspects of the bioeconomy is our understanding on how we can convert lignocellulose into simple sugars. So we know that this is controlled predominantly by microbes in nature, and we know they use a bunch of crucial enzymes to take this complex substrate into simple sugars. We also know that there are many natural ecosystems as well as engineered ecosystems that have evolved to do this process rapidly and efficiently. And these are the model systems that we use to try and create a deeper level of understanding to kind of confront this bottleneck. So because we cannot grow most of the organisms that exist in the natural world, we use a collection of what we call meta-omics. So these are different molecular toolkits that we can use to study the microbial communities that contain the information that we need to understand these processes at a deeper level. So metagenomics is the process of sequencing your entire DNA content within a microbial community. One of the big advancements in this area, and I'll talk a lot about this today, is the generation of reconstructed genomes from this metagenomic information. So these genomes that are assembled from contigs that have come from this metagenomic data. And these are called metagenome assembled genomes, or MAGs. So I'll be talking about these MAGs quite a lot today. And the reason why is because they really empower all the other omic tools that we can use to interpret the data that are contained within these systems. So when we talk about gene expression, we're talking about metatranscriptomics. When we're talking about protein expression and protein detection, we're talking about metaproteomics. So this is looking at the entire protein content in your sample. This is looking at the entire mRNA content within your sample. And we also use metabolomics. So we're looking at the metabolites that these microbes are producing in this system, and we're trying to incorporate all this information together. So this is where these genomes really come into power, because we can take all this gene transcript data, this protein data, we can map this all back against these genomes that came from the original organism that we're studying in the first place. So we can get much more information within individual populations that are found within our microbial communities. Okay, so that's the toolkit we use. We try and look at these microbial communities at different levels. We're interested in looking at individual enzymes that do the processes that we're after, in this case, conversion of lignocellulose. We look at the mechanisms that these enzymes are involved in. So it's very rare that these enzymes act alone. Quite often, they're found in kind of detailed mechanisms where you have multiple enzymes have multiple functions. We look at the microbes and how they express these enzymes and these mechanisms. We look at multiple genomes from a particular community and how they integrate with one another. So we look at the different microbial networks that exist in complex communities. And we look at how these different microbes coordinate this integration. So how do these organisms work together over time to complete a common task? So in this case, plant biomass degradation. So for this is population dynamics. And each one of these tasks gets more complex in terms of the community complexity. So I'm not talking about this is more difficult than this. It's more so that as we go further, we're getting into more and more complex communities. So it gets more data, more integration, more difficulty. Okay, so we use these tools, as I said, to look into microbial plant biomass degradation. We predominantly focus on known efficient animals such as herbivores. So we've done a lot of work on ruminants from domestic cows as well as more exotic species found here in the Arctic. I'll be talking today about some data from what I think is one of the first fistulated moose, at least that I've ever seen. We also work with insects, soil samples, important production animals here in Norway. So we're talking salmon, gut microbes, as well as different feedstocks that go in towards different production animals in terms of pigs. And we also work with anaerobic digestion that occurs in key reactors that supply energy to some of the major cities in Scandinavia. So some of you might recognise this as the Eggia plant. And what this does is it takes all the organic waste from the people in Oslo and converts that organic waste into methane, which then goes into fuel several of the buses, well over 100 buses that drive around the city. So this is an initiative from the Oslo Kommune to improve organic waste recycling. So these are some of the examples of the data sets that we work with. Before I get into too much data, I want to just quickly break down the plant cell wall, what these microbes are up against. So when we're talking a simple grass, in this instance, we see a complex matrix of cellulose. This is the most predominant polysaccharide we see in the plant cell wall. This is arranged in hard to degrade crystalline structure of beta-1,4 linked glucose chains. And depending on your plant substrate, you'll see a different variety of hemicellulose. So this is the kind of different types of polysaccharides that are gluing these crystalline cellulose fibers together. So this can vary depending on the type of plant that you have. So microbes have evolved to take advantage of these substrates. They use what are known as carbohydrate-active enzymes, or Kzymes for short. These are further broken down into different categories. So I'll be talking quite a bit about glycoside hydrolases. These are enzymes that'll break a specific linkage between two sugar units. We also have carbohydrate binding modules, which will bind to these different regions. We hear a bit about auxiliary activities. So these is where you find your lytic polysaccharide monooxygenases, which create oxidative breaks in crystalline chains. So when we look at microbes and their ability to degrade plant biomass, they all have, not all of these enzymes, but they all have several different types of these enzymes. And how they're used can greatly vary depending on the type of bacteria you have and the type of environment you're studying. So the first lignocellulose degrading organism described was an aerobic fungi. It was found to be chewing up GI tents in the Second World War. And it was found that this fungi produces two types of enzymes which will degrade cellulose. We have an endo-acting cellulase that will create chain breaks in amorphous regions in the crystalline matrix. And we have exo-acting cellulases which will processively work along these crystallized chains. In the 1980s, Ed Bayer and his colleagues identified that anaerobic bacteria have similar types of enzymes, but instead of being secreted outside the cell, they're arranged in complex cell wall attached structures called cellulosomes. In 2000, it was found that our human gut microbiota has a large proportion of gram-negatives called the Bacteroidetes. And the Bacteroidetes have outer membrane attached glycoside hydrolases connected to this typified Sus-C, Sus-D system. This is what we typify as a pool when we see these two lipoproteins. So this is a Sus-C outer membrane transporter. This is involved in polysaccharide binding. And we see different varieties of these polysaccharide utilization loci depending on the type of bacteria and the type of substrate that they're degrading. In 2010, Gustav, together with Vincent, discovered LPMOs at NMBU. So these are, as I mentioned before, oxidative, or we'll see data that will bring new information to that, but these are enzymes which will create an oxidative chain break in the crystalline regions and creating new chain ends for these exos to act on. In 2013, we saw again another mechanism being described where we have multi-modular enzymes. So these had both exo and endo-acting regions as well as carbohydrate binding modules, all contained within one very large enzyme, and were found to be very efficient in plant biomass degradation. Some of the gram-negatives, it's been found that this type 9 secretion system is crucial in lignocellulose degradation. And this is a system that will pump large enzymes out of the cell. Work that we published last year showed that certain bacteria with this type 9 secretion system will pump out very large multimodular enzymes which are connected with polysaccharide utilization loci. So you can see that the type of mechanisms we're dealing with are getting more and more complex. And this is just another one that we published last year looking at outer membrane vesicles. So most gram-negative bacteria, especially in the gut, are known to produce these kind of separated little mini drones, these kind of outer membrane vesicles. And Magnus, a researcher with us, was able to show that these things are loaded with enzymes and are able to degrade a very wide variety of different substrates. Okay, so that's everything we know from all different types of ecosystems. If we go somewhere like the rumen, we have quite a simplified view. This is because we can only grow a handful of bacteria from this system. So we know there's some that have cellulosomes, we know that some have polysaccharide utilization loci, we know that some produce outer membrane vesicles, and we know that there's anaerobic fungi there. We're not quite sure how they degrade plant biomass as yet. And this is because, as I said, we cannot grow most of the bugs that exist there. So in 2011, JGI published a huge metagenomic data set at the time from the rumen. They had 268 gigabases of shotgun sequencing data, and they constructed quite a few mags that came from this data. So we took the mags that were affiliated to the Bacteroidetes because we suspected them to be involved in plant biomass degradation via bioinformatic inference. And Adrian, who was a PhD student with me at the time, looked at characterising these two in particular in more detail. So we've published quite a bit on this AC2A, and today I'll talk about AGA. So AGA is a member of the Bacteroidetes. Together with Lindsay Solden and Kelly Wright in the Ohio State University, we did a broad phylogenetic comparison of this genome with all the available genomes that exist for the Bacteroidetes. And for this, we took this new data set, which was published last year in Nature Microbiology, which reanalyzed 1,500 different metagenomes and constructed 8,000 mags from all available sequence in NCBI. So what we saw is our AGA from the rumen fell within this, this novel family within the Bacteroidetes. So these are all uncultured representatives. These are all genomes from uncultured bacteria. We only saw genome representatives from the cow and sheep rumen, so this was kind of typified, localized to an environment. And we characterized our AGA in detail. So this is what we renamed Paraporphyromonas polyenzymogenus. So the reason why we called it polyenzymogenus was that this bacteria was producing, had a large inventory of multimodular enzymes. So these are all very large enzymes, multimodular domain. You can't see the CASI family here, but most of these are GH5s. So we do see different modularization. We see 2 GH5s in a hypothetical domain here. And this was the case for all these different substrates. We see all these cases of very, very large proteins with at least 2 or 3 domains. Most of them, or some of them at least, are hypothetical. When we looked at these multimodular enzymes, they all had the signature for type 9 secretion. And when we looked at the genome, this is a complete blueprint for the genome based on gene annotation of this complete genome. We could see components of the type 9 secretion system. We also saw that this bacteria had the ability to ferment sugars that came from this polysaccharide degradation, producing succinate and acetate, which is like key metabolites for the cow rumen. So because we couldn't grow this bacteria, we weren't sure if in fact it was capable of degrading cellulose. So Adrian took some of the GH5 multimodular proteins from this genome. This is actually one operon that contained 4 different varieties of endocellulases. We saw 2 within one at a type 9 secreted system enzyme here. We saw another here, multimodular, one with a hypothetical domain and several single ones. So Adrian had these artificially synthesized, expressed, and characterized the protein within. So what he was able to show is that all of these enzymes were in fact able to degrade some form of cellulose, some only able to degrade soluble cellulose, some able to degrade more crystalline substrates. We saw different products being produced, and we had crystal structures solved for several of them, which showed a binding, a narrow binding cleft, catalytic cleft, which was which goes with the biochemical data, which would suggest that these enzymes can only degrade linear substrates. So if you put hemicellulose against these enzymes, we don't see any hydrolysis. So it suggested that yes, this bacteria may well be capable of cellulose degradation in the rumen. So we were interested in, is this bacteria actually active in the rumen? So we know it's there, we see the genome, but are these proteins expressed and detectable in the rumen? So to answer this question, we collaborated with Matthias Hess and Lily Pasatolik from Pacific Northwest Laboratory in the US. And they generate a very large metaproteomic data set from the rumen. And so Leva, who is a postdoc with us at NMBU, she took this very large metaproteomic data set and she mapped it against all the available genomes that we had for the rumen. So we're talking about this 8,000 mag data set from which there's about 100 that are affiliated to the rumen. She took genomes from sequenced rumen isolates from the JGI, and she took these 5 fungal genomes which were just published last summer. So the rumen fungi are believed to play key roles, but up until recently there's only been 1 genome. So now we have 5, so we have a new data set we can compare this proteomic data against. So we're interested to see if our particular bacteria is active. This is 120 different populations that we found in our metaproteomic data set. These are all numbers of proteins that were mapped to them. So you can see this is 100 different proteins here, measured here. So we have 120 populations which are active. And the one that we're all particularly interested in, we saw about 150 proteins mapped to it. So we took these 150 expressed proteins, we mapped them against our map, our metabolic blueprint, and we could see that elements of the type 9 secretion system were being expressed. We saw a few representatives of these large multimodular proteins being detected in the metaproteome, and we saw the central glycolysis pathway being detected. So this bacteria is indeed active in the rumen. So in that slide I had 120 populations. How does our guy compare against the rest of the populations we see in the rumen? These are known cellulose degraders, Ruminococcus flavifaciens. We see about similar numbers of proteins being expressed. Fibrobacter succinogenius is another known cellulitic bacteria. We see several hundred. The big standout here was the anaerobic fungi. So this is hundreds, if not one of them is almost 1,000 proteins were detected in the metaproteomic dataset. And as I said, these have been suspected for a long time to be key roles in lignocellulose degradation, but until now we haven't really been able to detect their activity. So Leva's taken the detected proteins from these fungi, and she's actually shown that we see a large representation of all types of celluloses, especially the extracelluloses. So these are things that we've long understood have a key role, but we very rarely see them in metagenomic data and also metaproteomic data for bacteria. But for fungi, we see a very high expression of these extracelluloses. So it indicates to us that fungi is likely where the action is in the rumen, and it's something that we're actively kind of chasing up now. Okay, so I've spent the first time talking about how individual genomes, individual mechanisms. So what happens when we try and compare all the different microbes within a system? So for this, we've collaborated together with Lindsay and Kelly. So this is Lindsay. Lindsay's a PhD student who's done most of the work that I'm presenting now. So she did a large meta-omic data set on a fistulated moose. This is an Alaskan moose. And we've broken this down into several steps, so several different data collections. So we looked at metadata. What are the microbes eating in terms of polysaccharides? So we did a polymer microarray where we could detect all different types of hemicellulose, pectin, and cellulose fibers, so we can see high relative detection of all these types of fibers. We use NMR to see what type of sugars are being produced, so we could, which correlated well. We see glucose, which is what you would expect when you see high levels of cellulose and mixed glucans. They did a large meta-omic data set. So this is looking at what microbes are there, what genomes do we see. So Lindsay was able to create about 100 metagenomic assembled genomes. We see a lot of novel families, a lot of novel genera, and they've named a lot of these as well. She did metabolic annotation of all these genomes. So I talked about metabolic annotation of just one organism before. Lindsay's arranged this in a nice code where you're looking at polysaccharide degradation, you're looking at sugar utilization, you're looking at glycolysis, short-chain fatty acid production, and energy production. So these are all the different genomes that she's worked with, and she's looked at annotating all of these in one collective effort. So the one before showed us what they can do. The proteomics tells us what they are doing. So this is looking at expression of one particular mechanism. So this is looking at these pools, which we know are important in some of these Bacteroidetes, which are found in the rumen. So this slide here shows all these different genomes which are affiliated to the Bacteroidetes, and Lindsay's gone through and annotated all the pools that are found within. So this is a rough count. We see some of these have over 50 different, 10 to 15 different pools. The colour here indicates proteomic detection, so we can see which pools are active in this system, which populations are actively degrading the substrates that are presented to them. So how do we put all this information together? how do we combine the metadata, the genomics, the proteomics. So Lindsay created this really nice food web which illustrates which substrates are available, which genomes are abundant in terms of the assembly content coverage, which ones are active. So the proteomics here, these edges are connecting the active populations, and we can see certain populations that are really quite active. So these are what we call metabolic hubs. These are degrading 6 or 7 polysaccharides within our system and producing smaller sugars. So this gives us a food web of all the different microbes that are contained within this environment at this time point. Okay, so for the last 4 minutes, I want to try and take it a bit further and talk a little bit how we can look at these interactions over time. So how are these different populations coordinating this collective community function? So for this, we've worked with a biogas reactor that's degrading lignocellulose. And because this is quite a complex challenge to look at this type of data, we've tried to simplify a community from this. And what we've done is enriched one on cellulose. So what we have here is a sample from here, a media that's quite selective, and we're able to create an enrichment that has about 10 different microbial populations. It is able to take cellulose into methane. So it has all the required populations to create biogas, so like a simplistic biogas consortium. We analysed this over time, grew this over time. We did a meta-omic data set, so we looked at creating all the genomes that were within this system. We looked at looking at the gene expression, so we did transcriptomics on this. We did proteomics, so we looked at the proteins that were also being produced, as well as generating large amounts of metadata. So I could easily talk for half an hour about this, but what I'll just focus on is the Kzymes, so the enzymes that are degrading lignocellulose. So we took the 400 Kzymes that we could found that were being expressed by this entire community at this time point. So this is all the Kzymes that are being expressed over time, and the expression profiles that are similar. So this is not taken at an individual organism, this is taken as a total community. So these clusters represent Kzymes that were expressed exactly in similar patterns over time. This is another way of showing these 8 clusters here. So these are collections of Kzymes. As you move over time, they show similar expression profiles. So we see some are quite constant over time, we see some rapidly increase and then decrease. And when we look at the bacteria that are contained within these clusters, we see we have multiple populations produce Kzymes that have similar expression profiles over time. So we're interested in not only what type of bacteria these are, but what are the Kzymes that are contained within these expression clusters that have similar patterns over time. So these 4 in particular I'll focus on now because we see multiple organisms within these clusters. So I've arranged these 4 up over time. The first 2 here, we see similar expression profile at the early stages. When we look at the Kzymes that are found within these expression clusters, we see predominantly Kzymes involved in peptidoglycan hydrolysis. So these are targeting carbohydrates that are found within, say, the cell wall of dead or decaying microbial cells. So not exactly lignocellulose degrading Kzymes. When we look at this cluster here, which was actually one of the largest expression clusters, this is cluster 4, we see a large number of cellulosome and cellulases and hemicellulose. So this is what we would be expecting to see when we have a community that's degrading lignocellulose. When we look at the metadata, this coincided with this consortium's exponential phase, as well as the rapid degradation of cellulose. So this is what you would expect to see in a lignocellulose degrading community. Another interesting cluster, which was very large as well, which is this cluster 4. This was contained mainly hemicellulases, and we saw multiple species contained within this. And when we compared this against the metadata, we see this is up against at the stationary phase. We see this is towards the end of cellulose degradation, but this is also when hemicellulose is actually being liberated. So we see a large amount of hemicellulose being released and available to these enzymes at this time point, which would make sense why we see expression of the hemicellulose cluster coming after the cellulose cluster in this instance. Towards the stationary and end phase, again, we see the same 2 clusters that are quite high over the time. And this makes sense when you think that this cluster contains all the enzymes involved in microbial cell recycling. So scavenging dead cells, utilizing the carbohydrates that are found within. So this is stationary death phase. All other carbohydrates have been depleted. So we have these enzymes active on dead or decaying microbial cells. So one of the most interesting things that came out of all this was that we had one particular population which was made up of about 4 or 5 closely related strains, and this one population had elements within all these expression clusters. This was actually really surprising at the time. This population originally has been described as a protein degrader, not capable of lignocellulose degradation at any level, not suspected of polysaccharide degradation. And we see elements of this cluster and of this pangenome in all these different clusters. When we actually look at the genes, we see hemicellulases. We also see ones that would signify perhaps cellulases. When we look at these actual genes and these different strains within this pangenome, we see these have all come from horizontally gene transferred regions. So these are all genes that have been acquired to give this bacteria the capability to degrade all these different substrates that are present in this system. When we look at all the data sets for this population, we see that it's actually quite abundant globally. So it actually makes sense that this bacteria is so abundant in biogas reactors when you think that it has the abilities to degrade everything that is confronted with. Okay, so where do we go from here? So I've talked about different elements of microbial community analysis, individual organisms and their mechanisms, networks, as well as trying to look at this over time. This is just the tip of the iceberg in terms of the data, the complexity. What we really need to do is to integrate all this into one large metabolic food web so we can see how all this information is contained within. We also look at viral networks. We haven't had time to talk about this today, but there's all these other elements that we haven't even touched on today. So what about the fungi, the protozoa? What about permutations, dysbiosis, host genetic variants? All these factors have a huge interplay on how microbial community functions. So what can we use this information for? So what I like to give reference to is everything we know about the human gut microbiome. We know so much about that system in terms of what fibres we should eat, what foods we shouldn't eat, what fibres can do for certain populations. When it comes to important production animals, we know far, far less in terms of the bacteria that are there, the feed they consume, and how we can better manage those microbiomes. The same thing goes for biogas reactors. We put a lot of effort into creating these so we can take advantage of their ability to recycle organic waste. If we get a better understanding on the communities that run these processes, we get a better ability to predict, prevent, or even remedy problems that these systems may confront. So I'd just quickly like to acknowledge all the collaborators, particularly the NMBU, Vincent, as well as all the other PhDs and postdocs that have done this work. Hopefully I've acknowledged them all throughout the slides. The large network of collaborators we have, especially Kelly and Lindsay, a lot of the work I presented was work that we're doing together with them. And of course the funding agencies. So thanks for the time. Sorry.
Thank you, Phil. We have time for 1 or 2 short questions.
Thank you for the presentation. I have a question concerning Adrian's work, where you have shown that you express those cellulose-degrading enzymes individually, and I gather that you characterize them individually. So I was wondering whether you have seen their substrate specificity when you add them all together.
Yeah, so one thing I didn't go into detail is these multi-modular enzymes, particularly the very interesting ones, are incredibly difficult to express and to purify. So short answers, we've looked at synergy for these, and we haven't really had any clear answers if they do improve. Quite likely we don't have the right substrate. I mean, we're working with filter paper, Avicil. I mean, cows don't eat these substrates, so we're probably working with the wrong substrate if we're looking for synergy. So no, we haven't seen synergy as of yet together.
Yes.
Can you say anything else more about the hydrolytic vesicles that are secreted? How do they access substrate? Are the enzymes released? Do they take up? Do they have channels?
So we know that so the vesicles in Fibrobacter were discovered in the 1980s. They took photos of them. They know that if Fibrobacter is growing on glucose, you won't see vesicles. They're only found if they're growing on cellulose. We've found, I think from memory, it's well over 100 different K-zymes within these vesicles. They had very wide activity, so they were able to degrade all sorts of different hemicellulose, all sorts of different pectins. So Magnus did a lot of product analysis. If we enriched switchgrass, so we did pre-treatment of switchgrass with vesicles, and then we used a commercial cocktail to see if it's helped destabilize the substrate. We saw a 3 times boost in our commercial cocktail if we've pre-treated the substrate with these vesicles. So we suspect that these organisms are sending these ahead to remove the hemicellulose that's present, because fibrobacter will only degrade cellulose, will only live on cellulose, it will degrade everything, but it will only consume and metabolize from cellulose. So we suspect that it's kind of paving the road, if you will, so the bacteria can get access to the actual cellulose that it needs for its own metabolism.